If you're just joining us at 1:30, this is again a introductory tour of a tissue culture lab here at Seneca College in Toronto, Canada. Again, this is wide open to anybody on Earth that has the internet and has access to the channel. It's not a closed channel. If you are not a student in the class, please be professional in the chat. I would appreciate it. Okay? And I'll try and get to the chat when I can. I am streaming from my GoPro. It is strapped to my chest, so if I'm not standing right in front of my computer, I do not see the chat. So I will try and check the chat throughout, okay? Before I get started, let me just confirm that the audio and video is good. If you could just type that in the chat. Great. Again, there is a bit of a 10-second lag, so I'll be waiting for your... Excellent. Thank you so much. Okay. So let's have a tour. So this is our laboratory. Let's start with the most important part of the lab, the safety, lab safety. Okay, so you can see here we have all of our safety items: fire extinguisher, fire blanket, our spill kit here. We have our call button to security in case we run into problems. And of course we have our bin for our broken glass. Please put all broken glass in this bin. It's gonna happen this semester. Try to limit the amount of broken glass. Please don't cut yourself, but glass goes into this waste container. If we come over here, we can look at the sink. It's a standard lab sink, but on the side here, we have our distilled water. Turns on and off like this. Please make sure you turn off when you're done. Off to the side, we have some soap, 2 different types of soap. This soap here is for your glassware. This soap, the pretty pink soap, is for your pretty hands. That's how you remember it, okay? Probably use a refresh on that. So don't use this on your glassware because it's got moisturizer inside and it will have a hard time washing off your glassware. Don't use this on your hands because it is not meant to go on your hands. If we step back for a moment, come over here, we can see we have all our glassware here. Lots of Erlenmeyer flasks. We're not going to use any of those, but we are going to use our 250 ml beakers, every lab, and they're located right here. Coming across here, you can see more beakers, more flasks. We do have these pour buckets, okay? Looks like someone maybe turned on the hot plate here. They are made of plastic. We are going to use them to make up our solutions. So please don't turn on your hot plate. They will melt, and that did happen one semester. I was teaching and looked off in the side of the room, and black smoke billowing out from a student's bench. So make sure you don't turn on the hot part of your stir plate. I like to use these because they have a large diameter opening. We can put our powders inside, and they won't get stuck on the side like we would if we're putting them into a glass small flask. So make up your solutions in here, and then we're going to pour them into a graduated cylinder and top them up. If we step back here, other items here, we have lots of serological pipettes. Notice they are sealed, they are sterilized. So if you open them up outside the hood, they won't be sterilized. We'll talk about how to sterilize that a bit later. Looking down below here, we have lots of shelves of dilution bottles. So we have tons of bottles to make up your solutions. So you'll be mixing up your PBS and your MEM media in these bottles next week when you come to campus. You'll need 4, 2 for PBS and 2 for MEM. In these drawers here, well, hopefully I can't open that one all the way. You can see there's different colored caps. And we'll try and modify the color for each lab section to try and help you identify your bottle very quickly as compared to other sections. Over here, you can see we have a biohazard bag. So all of our contaminated plastics will be going in this bin, but never, ever, ever place any glass in here, okay? Glass goes in this beaker here. Oh, this piece of plastic I left out, so that would just go into this biohazard bag right here. Again, glass just goes in this bucket here. This would be your used past your pipettes. Please don't place your empty bottles of solution in here. For that, wash them, put them away yourself. Right here, you can see we have a large number of test tubes, and each one is filled with 4 cotton plug pasture pipettes. These are sterilized, so I will not open them, and you'll take one of these at each lab. So the ones out here, these are sterilized. Other things to note, well, here's the gloves. Here's your paper towels, and some Kim wipes are up here if you need them. As well, on this side, we have our standard sink, like on the other side, and we have, pardon me, and we have distilled water. Again, please turn that off when you're done. We have our pink soap for your pretty hands, and we have our commercial soap, but on this side, we also have a bucket or a container of bleach. I'll talk about that in a few moments. If I step back here, graduated cylinders, they're located down below here. Okay, you're gonna use these at your lab next week. You'll need a 1 liter and a 500 mil probably. And they're located down below. Please note, I see many a student using this to try and read the meniscus. That's not what this is for. This is a bumper. So when it falls down, it doesn't smash, okay? So please don't use this to read the markings on the side of a graduated cylinder. As we walk along, we can see a large stainless steel. This is a freezer, a standard freezer at -20. And this will be where you keep all your frozen stocks on these shelves here, okay? There's 3 sections, so I'm gonna have 3 separate shelves. Please note, don't put items in the doorway. The door is for me. You guys will have assigned shelves here. If we turn around, we can look down on the ground here. We have piles of buckets. This is a standard sort of bucket. This one looks like it's from Canadian Tire. And we're gonna use these by pouring in just a little bit of bleach and a squirt of soap and adding about 2 inches of water in there. It doesn't have to be distilled water, just tap water is fine. And all of our items, our plastic and our glass that touches the cells, we'll place these in here to decontaminate them, okay? So they're located just down below this device here. This device is called the Coulter counter. It's a particle counter. So you put your cells in here, bring it up, close the door, and over here we can press the start or stop button and it will suck up some liquid and count the number of cells in there. And we're gonna learn all about that this semester. If we come over to this side, we have the same unit here. It's a bit newer, but they both work well. I'll show you what happens. We press start. Hopefully you can see that there. If you're looking right there where the 0 is, you can actually watch it count in real time. 5, 6, 9, 10, 11, 12, 32. Oh, there, it just jumped a lot. Now, ideally, when this is clean, it should be below 50. Now, we haven't used this in a little while, so it's not bad, 48. So it could use it cleaning, but we won't be using this for a couple weeks, so not a big deal. If we come over here on this side, we have something called the Countess 2 FL. This is our automated hemocytometer. So hemocytometer, glass slide, we load up with some cells, and we can count that using a microscope. And we will do that this semester. The microscopes will be located below all the benches, as you've probably seen in other labs. Now, with the automated one, you see there's a little opening right here. We place our hemocytometer in there. The touch screen, we then press go. It will take a picture of the slide, and an algorithm will determine the concentration of cells in there for you. It's quite an amazing device, and you'll learn all about that this semester. If we turn over this way, on this shelf here, we have all of our chemicals. We like to keep them in somewhat alphabetical order, okay? We do have some balances here, both top loading balances and analytical balances. I have taken down all the chemicals that you're going to be using next week to make up your solutions. And Garija has graciously agreed to make up stations for all of you. So we can spread each other out and we can work together and not be delayed. So that's great work. If we come over here, well, I already showed you, we have our shower unit here. Stand underneath, pull that down. There's really no reason why we should be using the shower in tissue culture. The eye wash station, again, you put your face in there, you press on that, these pop open, it sprays your eyes. I don't think anybody has used this in tissue culture before, but it is here just in case, okay? Again, I did mention I have had to use it once before. I got some phenol in my eye, no damage at all because I washed my eyes out immediately. This large unit here is a minus, well, it's minus 85 right now, but usually we call it a minus 80 freezer. You can see it's quite large and it's very cold. Standard freezer is minus 20. And you'll be storing your cells in here. So I'm going to give you cells later on, and you're going to cryopreserve them, and you'll store them directly in this minus 80 freezer. And you can see I have my cells here. Here we go. Check this out. There's the cells I'm going to thaw next week for you. I got a lot of aliquots. I should have more than that, but I've been going through a lot over the semesters. This closes up, and that locks shut. Please note, you will not be allowed to go into this. Why? Well, many years ago, a student went in here and did not effectively close the door. And as a result, the whole thing thawed out. We lost all samples. So for the most part, this will be locked. If we come over here, there's one more item I want to show you. Right here. This is our swinging bucket centrifuge. So if I press the open button here, it will pop open. If you look inside, hopefully you can see this, we have buckets, and they sit on little hinges and they swing. So when it spins, this goes directly sideways, and your pellet will be on the bottom, not on the side, as you've used with a fixed rotor centrifuge. So maybe this is something new you're going to learn this semester. And the way you balance it is going to be a little bit more tricky. You have to draw a straight line through the middle of the rotor, and we'll explain more of that later on. Okay, so that is a tour of the main laboratory. I'm going to go back over to the chat and see if I can answer any of your questions. Any questions at all? Just do type them in the chat. I don't see any questions. Okay, so let's continue on with the tour. Going into the main lab, the side lab here, you can see we have these big units here. These are called vertical laminar flow hoods. Laminar is the opposite of turbulent. So it's going to suck air into the vents at the front and the back, draw it up the back of the device. There's big blowers inside. There's a giant HEPA filter right here, and it's going to blow the air through the HEPA filter, sterilizing it. And it's going to create laminar flow. It's going to be like little columns of air, and will keep things sterile. You will be assigned one of these for the whole semester. Let's just pick this one here. To turn it on, this sash is very much like in a fume hood. This is not a fume hood. We open it up, and you'll notice, hopefully you can see that right here, there's a little line right here. So you want to open up about to that height. If we go to the other side, there's a button here that says blower. When I press that button, watch the screen right there. Do you see what happened? It says, please wait. You want to see that. This means that I have effectively touched the button and the machine is starting up. Sometimes students don't push the button hard enough and that symbol doesn't come up. That means the machine will not be working. It will not start up the blower. You won't hear it because there'll be 11 other hoods running. So make sure you see the please wait symbol. And for that, we must wait before we can sterilize the inside of our laminar flow hood. And maybe you can hear now it's getting a bit louder. Some background noise, some airflow. So this is the working surface you're going to be working with. Let me pull this out for a second to show you. So you can see it's a big stainless steel plate. This is the thing we want to sterilize. If you look underneath it, it's just empty space. We do have some shells there from our primary culture from last semester. But you're never going to take this out unless you spill chemicals in there. So it should just look just like that. Okay. If I look down here, there are two stools, a tall one and a short one. The short one is for you to sit on. The tall one is to put your items on. And to sterilize this, we're going to use 70% ethanol. Down below here, 70% ethanol in a spray bottle. We can't sterilize it, and oh, the symbol please wait did come off, so that it is ready to be sterilized. I do want to show you one thing before we sterilize it. Down here along the side of the incubators, well, here are our 37-degree incubators. They look like a standard incubator. However, CO2 tanks connected to them. We'll come back to this in a few moments. Here I'm going to get a garbage bag. This is a standard Costco-style garbage bag. And we're gonna tape it to the side. Now I'm right-handed, so I'm gonna tape this to my left side so I can drop items into it. If you're left-handed, you'll do the opposite, okay? You get your masking tape, whoops, rip off a piece, and tape it right to the edge. Now, when we're working in the hood, our hands are going to be sterile. We don't want to we don't want to touch the bag. So what we want to do is we want to make this so it stays open. We can fold this over and we can tape it up like so, in which case it will stay open. Again, there's a whole YouTube video I have of this, but I thought I could show you in real time, okay? Let me just go back to the chat for a second and see if there's any problems, because I don't know what's going on. I'm assuming this is all running smoothly. All good, guys? Sound is good, audio, video is good, no problems. Okay, great. Let's go back to it. I just wanted to make sure I wasn't talking to nobody. I am just gonna take one of these buckets. Again, we would normally have a bit of bleach and a bit of soap and about 2 inches of water inside, but for now, I'm just gonna use it as empty. So I would place that on this side because I'm right-handed and I'll be taking items off and dropping them into these. If you're left-handed, you'll tape it to your left side so you can take items up and drop them inside. Now let me show you how to sterilize this hood. We're gonna use 70% ethanol. We're gonna take a piece of paper towel. We're gonna fold it up like so. 70% ethanol. We want a nice diffuse spray, like that. Don't have it shooting out. See if I can do it. There you go. We don't want that. We don't want to have a gun shooting. We want a nice diffuse spray to try and get the little droplets of 70% ethanol everywhere. We'll then reach inside and wipe back and forth, moving towards the front of the hood, pulling contaminants out of the hood. And we'll place that into our bag. In which case then, we're ready to work. You can see we're ready to go. However, when I place my hands inside, make sure I spray them. Also, make sure you take off any watches you might have. Source of contamination. And now that I'm inside, hopefully you've been practicing your exercises where you're sitting up straight, arms up, because you can't have your arms covering these vents. If I do that, watch what happens. It alerts me, airflow problem. Also, if I open this too high, an alarm goes off. So always make sure you have this at the proper sash height. Other than that, I'm not gonna do anything else with this. When you're done, you'll close the sash, you'll take off your bag, close the sash, push in your stools, put things back where they came from. I'd like to keep the lab very organized. Please maintain that as well. We'll then bring this next door, our bag and our bucket. The bag has only paper or plastic that's not contaminated. Never any glass. That goes in regular garbage. Our bucket here, we did decontaminate our items inside, but I would still take out all the plastics and put it in this bin. I would take out the glass and put it in this bin. And then we will autoclave it afterwards. I then rinse out my bucket with some water and place it back down below there. Okay? Let me go back to the chat and see if there's any questions or any problems. Everything looks good. Okay, let's keep going. So that was a walk through the laminar flow hood. Let me show you some other things in this lab. First up, on the top here, we have these test tube racks. We have large holes for 50 mils, 15 mils, and some smaller Eppendorf tubes. These have to be sprayed with ethanol as well. And when you spray them, make sure you spray the outside and the inside. Get the whole thing sterilized. As well, don't spray these with test tubes inside. Spray this separately, spray the tubes separately, put them into your hood, and only combine them after they're inside the hood. Up here we have more gloves if you get a rip. We have 15 mil and 50 mil conical tubes. If they're sealed, they're sterile, or if they're taped shut, you can assume that they are still sterile. They're still good to use. Down below, we have 10 mil and 5 mil serological pipettes, okay? And this is mainly what we're gonna be using. I see a lot of students wasting time in the lab. You know, to open these, you can go in and you can get in here and you can try and peel it open. Right, you got this little thing, you can try and peel it. That's a waste of time. Best way to do that, there's plastic on this side, paper on this side. Take this and just push it up that way. You can see it rips through the paper side. I can then take a pipette controller, stick it in there, and then you would rotate this so that the numbers you want are facing you. Then you would take this off, you'd use it, you'd take this off when you're done, and drop it in your bucket for decontamination. I guess next up, down here, are pipette controllers. I'll discuss more of this later on in the session. They are charged wirelessly in the dock, so when you're done, please do put them in the dock. And at the very bottom, we have our spray bottles of 70% ethanol and some paper towel rolls. Coming over here, we have two, just get rid of this. For now, it's not dirty, so it's not a big deal. We have two inverted microscopes, okay? You can see, you look here, it has your, I don't know if you can see this, your objective lenses are on the bottom and your light source is on the top. And if we turn it on, the on button is right here. You can see the light comes from the top, not from the bottom. Let me see if I can turn this a bit more. There we go, that's maybe a bit better. So here's your objectives and here's your light source. It is binocular, so please utilize both lenses. If you don't know how to use them properly, I will teach you in lab. We have our fine adjustment and coarse adjustment knob and our light adjustment. And on top here, we have it connected to a monitor. So when I'm looking at cells in the microscope, I can look up. You can all see what I'm looking at on the screen. When you are done, please do remember to turn off the light. Let's not waste energy or waste our supplies. That bulb will burn out, even though it is LED. Here we have a whiteboard. It's empty right now. But for most of the semester, I'm gonna write the protocol for subculturing on here. So that will remain on the board for most of the semester. At the end, I will erase it because you should know that. If we look up at the top here, you can see we have a clock. It has a second hand, that's very important. You're gonna watch that go around twice because we're gonna be incubating in trypsin in our incubators for 2 minutes. So you gotta time that. So you'll be spending a lot of time here staring up at the clock, waiting for 2 minutes to pass. If I come over to this side, we have a refrigerator. You're gonna place your PBS, your trypsin, and your MEM in here. These will be your working stocks. Please only use the shelves. I'll have a shelf for each section. Do not use the door. The door is for me. Please and thank you. You're gonna have other supplies, and they're gonna be stored in the cold room, and I'll show you that in a few moments. If I turn around here, oh, we can see there's a secret hood over on this side. Not secret, but it's just out of the way. We do have our CO2 tanks. You will not touch these. These are run by our very competent lab techs. We do have 4 incubators. They look like a standard 37-degree incubator. Don't know if you can see it, but up top here we have the temperature and we have the concentration of CO2. So we're pumping in an atmosphere of 5% CO2 into those incubators. If we open the door, it opens just like a standard incubator you've had micro, but there's a secondary door inside, okay? And it is sealed. There's an airtight seal around there. To open this, I take this and I rotate it. I can then open this up. You can see there's a rubber gasket here that creates an airtight seal. As I keep this open, I'm not sure if you can see this, I'm trying to lean back. Maybe you can see the temperature drops and the CO2 level drops, right? So please don't keep the door open too long. Open the door, do what you need to do. There we go, we can see the temperature has dropped and my CO2 is dropping. When you're done, make sure you close the door, turn the knob so it is sealed. And then when you close this, make sure it's fully sealed. I see some students sometimes just closing the door like that. Please don't do that, okay? It did close this time, but sometimes it closes and it hits and it bounces back a little bit and it's cracked open a bit. If that's left over the weekend, This is a heated door here. If this is open, the temperature inside and outside of the glass will be different. What you'll get is condensation filling the entire inside. We do not want that. So when you're done, please make sure you do close it. One other thing I'll point out, see the numbers? Each of you will be assigned a number. So if you're assigned number 4, you get this part of the incubator for the whole semester. This little bit from the front to the back, okay? So there are 30 of you, so we will have to share the incubators. If I come over here, there's some other items here. When you're done with your pasture pipettes, you're gonna reload them. We don't reuse them, we throw them. When we're done using them, we put them into our bucket of bleach. You come back over here, always load up 4 more. And when they're done, a lab tech will take these and autoclave them and place them back in the other room. So as a reminder, please don't use these in the lab. These are not sterile. The sterile ones are next door. Okay, I think that's all I wanted to show you in here. Let me go back to the chat and see if there's any questions. No questions. Okay, you're already ahead of me on this one. Okay, great. What I want to do now is I want to walk you through how to use a pipette controller effectively. Okay? Let me just move this stuff over here so I can actually see the feed and maybe answer some questions as I go along. Let's move this over out of the way. Oops. That's not good. Okay, so I can see the chat, so if you do have questions, I can answer them in real time, hopefully. So what I have here now, I have a pipette controller. I have a 5 mil pipette, serological, and a 10 mil serological pipette. I have a beaker of distilled water. And I have a paper clip that has been opened up. Nothing fancy about it. Okay, so let me explain to you how to use the pipette controller. So, pipette controller is just like a gun. You hold it in your dominant hand. I am right-handed, so I would hold it in my right hand. Let me just adjust the camera a bit. There we go, maybe that's better. Okay, I think that's better. Let me just, it's a bit of a lag. Let me just make sure it's in the field of view. Yeah, that's better. Okay, so you can see here, there are 2 buttons. If I press this button here, it sucks liquid, or it sucks air up, and if I have an airtight seal, it's gonna suck liquid up. This button here, if I press that, it blows air out. So we can use this in the lab instead of those silly pipette bulbs to effectively use our pipettes to transfer liquid. Now, if we look at the top here, there's a screen, there's 2 buttons. Well, just give me a second to make sure you can see the screen effectively. Try and bring it a bit closer. Maybe you can see that. Yeah, it looks good. So right in the middle, you can see that there is a battery. So this tells you how much charge is there. This one is 100%, so it's been charging the whole time. Up here, we have a button. This button, if I press it, let's see if you can see that. I'm not sure if you can see that. It switches between blowout and EX. Yeah, you can see that. So you always want to have the device on blowout, not on EX. So let me show you the difference between the 2. Let me open up this pipette here. Again, I'm going to push it through the paper part. Rotate it so I can see the numbers the way I like, and then pull this off. I will then submerge this into the liquid, and let's say I want to do 10 mils. So I will draw some up and then bring it down until my meniscus touches the 0 mark, right? From 10 all the way to 0. I'm counting backwards. Oh, it looks like it's dripping a little bit. Maybe don't have an effective seal there. When I'm done, if I want to dispense this, I can press the blowout button. And you can see it sprays out the liquid. As we do with a serological pipette, we blow out the last drop. Now, if I switch it to EX, you'll see what happens. I can suck up exactly the same, get 10 mils. But now when I dispense, watch the difference. It's just opening up the area and letting gravity pull it down. So it does take longer, and you'll notice the last drop remains in the tip. As a result, you would be using this for a volumetric pipette, which we will not be using this semester in tissue culture. So please set your pipette to blow out at all times. Other buttons to notice here, there's one on the bottom, and maybe, hopefully you can see there's a graph right here, a 1 through 5. This button on the bottom here controls the speed. So if I press this, you can see it goes 1, 2, 3, 4, 5, and then I press it again, it goes back to 1. So 1 is the slowest, 5 is the fastest. Now this 1 button controls the speed of both our suck up and our blowout buttons. To adjust it, what we do is if I want to adjust the suck up, I make sure I engage that one first. And now I can adjust the speed of this. I always set mine to 4 for suck up. This button on the bottom here, I want to adjust the speed of that, I engage that one. Now this, you can see it was at 4, now it's at 1 because it's a different setting for the lower blowout button. I always have this one on 5. I like maximum speed to blow out, and I like to have it a little bit slower when I'm drying up. When you are starting in the lab, I would like you to have yours lower until you get better at using this device. So once again, submerge it, bring it up, measure it, and dispense. If we switch to a 5 ml pipette, the diameter is smaller. What you'll notice is the liquid rises a lot faster in the pipette because the diameter is smaller. So what you may want to do is you might want to lower the settings. When you switch to a 2 ml pipette, that's very fast, or a 1 ml pipette, I even lowered this down to about a level 1 or level 2 because you do not want to suck up liquid into your pipette controller. What do I mean by that? Well, if I start sucking up and I'm not paying attention, this keeps going up and up and up and up and up. And if I keep going, you'll suck liquid up on the inside. That is very bad, okay? So you should try and pay attention and avoid drawing liquid up in there. It might happen, so I'm gonna explain how to fix that right now. If there's any questions in the chat, please do throw them in. So, what happens is there's a little valve inside. That's a one-way valve. If water gets sucked up inside, it presses on the valve and seals it up. So if you're working away and all of a sudden you can't suck liquid up, stop what you're doing. In all likelihood, you have sucked up liquid into the pipette and the valve has stopped it. If you keep sucking liquid up, it's eventually the valve will fail and you'll suck liquid up into the controller and probably destroy it. We have not had that happen here in the many years we've been using these. So I expect you all to work and maintain the use of these. Now, if that does happen, there's a simple way. We have to take it out, clean it, and dry it. Okay? This right here, it unscrews. So make sure when you unscrew it, you have it over a bench because there's little items inside that can fall out and sometimes be lost. So I just take this and I unscrew it. Okay? And inside we have a little sterile filter. And if I turn this upside down, we have a gasket to hold the pipettes, and we have our valve right here. And if I shake it, hopefully you can hear that. It's a rattling sound. That's what you want to hear. If it rattles, that means it is open. If it doesn't rattle at all, you do want to make sure you open it up and make sure you clean it. Okay? So I need some paper towel. 1 second. I have to grab a roll of paper towel. There we go. I have to go all the way back. Oh, I'll just grab this one here. So if we get media inside, this could be contaminated and stuff could grow inside. So not only do we have to get rid of the water, but we have to get rid of any media inside. So what we're gonna do, we're gonna open this up. If you look at the bottom, we can see there are little holes in the bottom. And on the top, we have this black gasket. Hopefully you can see that. So what we do is we take our paper clip and we press it through that hole and press down. It's gonna pop the bottom open. Be careful, little items fall out. So just hold on to it, take it apart. So if you look here, hopefully you can see that. There are 3 parts to it. We have the top part. It has that little rubber gasket at the top, and inside it has a larger rubber gasket. We have the bottom part. Actually kind of looks like a little petri dish. Little holes in the bottom. And we have this little valve here. And hopefully you can see this in the camera. There is a large diameter and a small diameter part. Can you guys see that? Oh, let me just get into the black a bit. Again, there's a bit of a lag. No, gotta go further down. Can you see that? There's a large part and a small part. Now, there could be media on this, so to clean that, we take the items and we drop them in a little beaker of distilled water. Swish them around a little bit, and then take them out. It's just distilled water. Don't lose these. Okay, now we must dry them. They must be completely dry. Any water in there at all, the second you use it, it's gonna seal up that valve and you're done. So get a paper towel and effectively dry every little bit of water on all those 3 pieces. Make sure you don't lose one of the gaskets inside. They can come out, so make sure you don't lose them. I like to just make like a little drill bit there and kind of stick it in there to get that dry. Completely dry. Again, if there's any water, it'll seal right back up again. Let's put it back together. So remember, we go back to this little valve, the large part on the bottom, the small part on the top, and we just put the small part up into the inside there, like that. So I don't know if you can see that, but the small part is pushed up against the rubber gasket and the large part is on this side. I then take my little Petri dish and just cover it up there. The flat part is there. So this should be clean and dry now. And if you shake it, hopefully you can hear it rattles. Can you hear that? Can you hear it rattle in the camera? I don't know if you can hear that. Anyways, yeah, okay, great. Again, there's a bit of a lag, about 10 seconds, so that's why it's a bit weird. Now, sometimes when you use this, you'll come in after the weekend, sometimes you'll be rattling, you'll shake your controller to see if it's contaminated. And if it rattles, you're good. If it doesn't rattle, it doesn't mean it's contaminated. Sometimes that little valve inside just sticks. So you just have to take it and just give it a smack. Smack it on the bench. And then give it a shake, and this should be good. Now, when we put the rest back together, here is your gasket for inserting your pipettes. Notice it is a cone. There's a large part of the cone here and the small part over here. If there's any water inside, that's gonna go right up inside and contaminate. So we have to dry this as well. To do that, again, I make like a little drill bit and I put it into the large part of the cone and just kind of rotate it and get it all the way in there. Make sure you get all the water out. Again, if there's any water in there, the second you put it back together and use it, it'll seal that valve up again. So get that completely dry. So we have our valve, or our gasket, we have our valve, and we have our sterile filter, okay? So this is a sterile filter. Maybe you've used one in another lab with a syringe to try and sterilize a compound. That's what this is. This is our last line of defense. So this is inside, it's kind of set up like this, where you have your gasket, then you have your valve on top, and then you have this sitting on top of that. So if your valve does fail, if you keep sucking, you draw liquid up, at least this will prevent microbes from getting inside the pipette controller. It's not gonna stop water, but it's gonna stop microbes. So please, please, please pay attention to what you're doing. Now let's reassemble it. So I take this bit and I take my gasket and I put it so the large part of the cone is facing downwards. So the large cone is here, small part is up top. I then take my valve and I place it inside, right on top of the gasket. So I can see the word top and I can see the small gasket there, the small rubber gasket is pointing at me. I then take my sterile filter and I put it in like that, so the knobby bit is pointing upwards. And then I take this and I place it on there and I just screw it tight. And again, when you shake it, you should hear it rattle, okay? So if it rattles, that's good. If it doesn't rattle, it could be it's just stuck. Take it out, give it a smack. If it is contaminated, please do clean it, okay? That's all I had planned for today. If we were in person, you would be practicing. I would have you working on this and cleaning out your stuff. I did try something new today. I tried this about a year ago, but had some problems this morning. I think this is much better than perhaps me sitting at home in my office talking at you. But if you do have questions about anything today, please do type them in the chat. Do we have to clean the controller every lap? No, great question. Ideally, you take responsibility for this, okay? If you contaminate it, it is your responsibility to clean it. In the past, we actually had numbers on them, and if someone didn't clean it, then we go after them. Please, let's just agree as a group that if you contaminate this before you leave the lab, please make sure it is clean, okay? That way, the next person who comes in does not have to clean this. Now, it does not have to be cleaned every lab unless it is contaminated. When you place this in the hood, however, you will spray this down with ethanol, so you'll be sterilizing it every lab. But cleaning it should not be necessary because the person before you should have made sure that they did not contaminate it. Great question. Any other questions I can answer, guys, while we're here? Again, I apologize, there's a bit of a lag, so there might be some dead air between. Okay, so a few reminders. There were some changes here at Seneca for the rest of the semester. Those changes do not affect any of you. So third year lectures and labs will be in person starting on the 24th, okay? But the remainder of this week will remain online. So lecture will be on Thursday, online, and lab, I have some calculations to do with you. I'm gonna review cell enumeration. But next Monday, Tuesday, you'll be coming in and you'll be doing all your solution prep. So again, I have all of your solutions lined out here. I took photos of them just before I started this. And when I'm done, I'll go to my office and I will make maybe a long PDF, a page for each one, and then you guys can start your stoichiometric calculations. Please note that these calculations are due before, let me repeat, before 9 o'clock on Friday, okay? This shouldn't be hard. I did the first calculation with you, just follow suit. There should be 14 calculations total. Are there anything else I can answer for you guys while you're here? Nothing else? Okay, well, I hope maybe this is your first time on Twitch. Maybe not. Maybe you're a bit of a gamer and you watch people on Twitch. But I thought it was a cool platform, and I have been on this just a couple times. But I'm real happy with how this went. Hopefully you liked it as well. I'm going to watch it myself again later on and see how it went. And it's a good platform.